Hi, good morning, student. Today we are going to discuss a very important topic, okay, that is science and the scientific method. Before we move to the scientific method, it seems convenient for me to explain the difference between the science and philosophy. You know very well that science, is the knowledge which is based on observation, experiment, and finally result or conclusions. Okay, but if you are going to study the philosophy basically based on only the logics. Okay, it does not focus on the experiment. Means whatever the philosopher says, okay, he cannot prove in the scientific laboratory, but whatever the scientist says, he can prove it in the laboratory. Okay, so science, is a knowledge which based on observation, and observation, very well that we have five senses. Okay, with the help of five senses we observe the thing. Okay, then we perform an experiment. Okay, and we collect the data, and based on observation, experiment, and data, we analyze the situations. Okay, and then we give the theory or laws, okay, or conclusions. Okay, so now let me to just move to the scientific method. What are the particular steps, okay, which are required for the research work of science? Okay, so let me to share my slides. Okay, yeah, you can see. Yeah, here you can see the slides. This is the first slide. Okay, now you see here, welcome to the scientific method, the steps that we have to discuss, okay, regarding the science or the scientific method. Okay, it's there are, there are some particular steps. Okay, let me just to open these steps. Okay, yeah, see here, if you are just going to study the scientific research method, okay, the first thing that you can see here is the to ask the question. Okay, first you ask the question. After that, you have to you have some background, okay, for that particular research. After that, you have to make an hypothesis. Well, now what is hypothesis? Hypothesis, is the guess that you can just guess something to solve the problem. For example, if you see that the clouds, black clouds are coming. Okay, you may guess that it might rain. Okay, so after the observation, okay, we construct a hypothesis to solve the problem. Okay, either our hypothesis is correct or not, okay, we have to perform an experiment. With the help of experiment, we just collect the data. Okay, and based on the data, okay, we analyze this data. Okay, and then we give the conclusions. Okay, either the hypothesis that is our guess, okay, is true or not. Okay, after that we make the result or the conclusions. Okay, these are the some particular steps of the scientific method. Now come to the the particular definition. Okay, what is the scientific method? It's a process that is used to find answers to the questions about the world around us. Yes, actually, when we see in our surrounding that something happening, okay, so the scientists always try to find out the reason why it's going on. Okay, what is the reason behind it? For example, if the light illuminated our room, okay, what is the phenomena is going on? If the fan is just moving, okay, what is the mechanical process that's going on? Okay, so scientists basically see around around the surrounding. Okay, and based on the observation, okay, he asked a question and then try to answer these questions. Okay, so this is the base of scientific method. Now, is there only one scientific method? The question is there only one scientific method? If you're going to study the scientific method, you'll find there are so many so many scientific methods. Okay, but if you are going to summarize all these scientific methods, you will see that in all scientific method, the first thing is identification of a problem, means what the problem is. Okay, what the problem is. Okay, or you ask the question when you see your surrounding, you ask the question that what's going on. Okay, and then you try to answer it. Okay, so you ask the question, you observe the thing, and based on that you are going to just perform an experiment, and based on the experiment you find out the facts, that's what's going on. Now, they provide an organized method for conducting and analyzing an experiment, means whatever the experiment is done, okay, it has to be very organized. Okay, for example, if I say the boiling point of water is 100 centigrade, okay, now I can go to the laboratory, okay, and I can prove it with the experiment that the boiling point of water is 100 centigrade. Okay, when I'm performing experiment, okay, I have to be very much organized. Okay, so now come to the next one. What is hypothesis? Yes, as I've told you that hypothesis is a simple guess. If you see the clouds, okay, you may predict that it might rain. Okay, so what is hypothesis? It is an educated guess based on observation and knowledge of the topics. You state as it is possible, answer the question. For example, see here in the diagram, you can see a happy dog will eat more, yes or no. Okay, see, if if the dog is very much hungry, okay, so of course he's going to eat more. Okay, but we can guess by observation that what is the condition of the dog. Okay, based on the condition, okay, we can predict, we can guess that he either the dog is going to eat more or less. Okay, so now mode of effects, the appetite of dog. Okay, it eat more or eat less. Okay, so this is we call as a two-tailed hypothesis. This is one-tailed hypothesis, this is two-tailed hypothesis. In one-tailed hypothesis, mostly we are asked the question to ourselves, yes or no. Okay, and in two-tailed hypothesis, we ask the question either the dog is going to eat more or less. Okay, now another thing, see, straight in the if and then. Yes, if it happens so, then it will happen so. Okay, so see here, if and then format. For example, if I water three plants with a different sodas, sodas, the cold drinks, carbonic cold drink, carbonated cold drinks. So does then the plant that receives sprites will grow the tallest. Okay, means if you are going to add a sprite, okay, in the plant, so it's going to grow tallest. So what is the reason behind it? What is the reason behind it? If you are going to add some Pepsi, okay, or 7 Up, okay, it doesn't grow faster. But if you are going to put some sprites, okay, so it's grow faster. So what is the reason behind it? So actually, scientists also make some type of statement or format that if it happens so, then it will happen so. Okay, now for this, the scientists just perform an experiment, and based on the experiment, okay, scientists collect the data. Now what the data is? Data, you can see here, it is information gathered during an experiment. Whatever experiment you are doing, okay, you have to gather the data, you have to gather the information, and this information has to be written in the form of table, and based on the table we have to make the graph. Okay, so we have to make different type of graph that we are going to discuss later. So what is data? It is organized into a data table and displayed visually as a graph. Yes, so whatever the data that we have, okay, we have to organize this data in the table, okay, and based on the table or the data table, we have to plot a graph. Now different type of graph is there, as you can see here. Number of students are there on the x-axis, and the y-axis, there are, like, some juices which are having a different colors. Okay, so which color is the most favorable color for the student. For example, if you are going to study in some particular area, and we give, like, the different color of juice. Okay, so how many students, like, take the red juice? You can see here the number of students only two. How many students take the orange juice? So there are, eight students who take the orange juice. And how many students take the yellow juice? So see here, this is the highest number. This is the highest number that the student select the yellow juice. And purple, is, you can see, intermediate. So this is the bar graph. Okay, based on the bar graph, okay, we just find out the personal likeness or dislikeness. Okay, or the we just predict, like, that what is, like, the favorite, what is the favorite color of the juice for the students. Now come to the next slides. Yes, graph. See here again, the bar graph is there. You can see here the bar graph has growing occupationals. Okay, so see home health aides, okay, human services workers. Okay, you can see here these all these, all these are written in the x-axis. Okay, and the y-axis, you can see on the y-axis, the percentage is given here. For example, 138% is for those peoples who have home health aides. Okay, and human services workers, 136%. And the lowest one, you can see here, medical records technicians. Okay, so here we just, like, plot the bar graph. Okay, based on the percentage and their occupations. Now another, you see here, another graph given here, our favorite sports. Yes, number of students. Okay, and what is their sports? See here, for example, saucer, is the most favorite game or the sports for the student, because you see here how many students selected this sports, about 9 students. Okay, and then here, basketball, only just 6 students. And here you can see the other games might be cricket, okay, football, badminton. Okay, the other one, you can see here, it's about three students. So when you plot the graph, you see here, have an axis horizontal. X-axis mean horizontal. This is x-axis. Okay, or if you are going to see here, this is x-axis. On x-axis, the professions names are mentioned. Okay, and on the y-axis, what is their percentage that is mentioned here. Okay, and here number of students are mentioned on the y-axis. So whenever we plot, like, the bar graph or the line graph, we have to make the two axes. We have to make two axes, x-axis and y-axis. Now the x-axis has a number for the amount of the stress being measured. Good when you are plotting data that spans many years or days, weeks, has really big changes from year to year or day to day, or when you are comparing the things. Means when you are comparing the things, okay, you have to use this graph. Okay, now line graph is another type of graph. You can see here, the same as I've told you that in the line graph, we have to make two axes. Y-axis is there, and this is x-axis. On x-axis, you can see the days are given here. Okay, and here on y-axis, the facts I got correct. Okay, how many, like, persons are there. Okay, and another graph you can see here, that on the x-axis the days are there. Okay, and the y-axis degrees in Fahrenheit. For example, like, if you are given the task to measure the temperature of different areas. Okay, so see temperature in the New York. Okay, it varies. You can see here. Here you can see day one. On day one, basically see the temperature is 43 Fahrenheit. Okay, and day two, 53 Fahrenheit. Okay, and then you can see here, this is day number three, this is 50 Fahrenheit. Okay, so as you can see, as you are just moving, okay, from day one to day six, the temperature gradually increasing. Okay, so see, here can be used when you are plotting data that has peaks ups and valleys downs, or that was collected in a short time period, used for two sets of numerical data. For example, time and the temperature. So time should be taken here, on the x-axis, and temperature should be taken on the y-axis. Let me to explain you one more thing here. Whenever you plot the graph, okay, you have to be very careful that the independent data and the dependent data, I will discuss this data later on, but I just want to tell you here, on x-axis we always take the independent data, independent variable, and y-axis we takes the dependent variable. Okay, now come to the next one. See here, steps of our scientific methods. See, choose a problem first, as we discuss it here, first we have to choose the problem, what the problem that we are facing. Okay, and for example, before 2 3 years ago, okay, there was a pandemic, okay, corona, a new virus, new virus was introduced. Okay, and this new war, just because of this new virus, so many people were being killed, okay, were being died. Okay, so see here, so the what the problem is, the problem was that at that time is the peoples are dying, okay, because of the virus. Okay, after that, the scientists did some research, okay, and they made a specific vaccine that is responsible to kill the virus. Okay, and for before this scientists also make hypothesis, either the peoples are dying because of some virus, or because of some poisonous gases. Okay, so then after experiment, the scientists came to know that the real cause of the death is the virus, that is the coronavirus, very well. So the first steps to choose a problem, what the problem we are facing here. Okay, secondly, do the research, okay, or do observation around, okay, what's going on. Okay, and third, you have to solve this problem. Okay, so for this you have to make a guess or develop a hypothesis might be three or five, okay, maybe sometime two. Okay, so after that, if your hypothesis is correct or not, okay, you have to perform an experiment, either your hypothesis is correct or not. Okay, so which, for example, if you select, if you make five hypotheses, okay, which hypothesis is correct. Okay, so this can be proved by the experiment. Okay, experiment is going to prove that which hypothesis, which one, or which guess, okay, is correct, or which one is wrong. Okay, now come to the next one here. See here, yes, again you can see summary. Basically we are moving to the to summarizing our topics. See the first one, choose a problem, state the problem as a question. Okay, means you questions in the problem. Okay, secondly, research your problem. Yes, get 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 advice and make observation. Okay, first you observe the problem, then you make observations. Okay, and after that, based on your observation, you develop hypothesis, you develop some gas to solve this problem. And after that, if the hypothesis or your tentative guess is wrong or right, okay, you have to perform an experiment, design an experiment, plan how you will test your hypothesis. Okay, and then after the experiment, you come to know that which hypothesis is justified, Okay, test your hypothesis, conduct the experiment, and record the data. Yes, when you perform an experiment, of course, like you find so many data, you collect so many data that has to be tabularized. After that, you have to make the graph, okay, maybe the line graph, maybe the bar graph, okay, then organize your data, create a chart or graph. You can see here in the sixth step. And seventh, draw conclusions. Yes, what is the result? What is the result? Okay, after experiment, okay, which hypothesis is justified? Okay, this is our conclusion basically. Okay, so this is these are the steps of scientific method, and this is a summarized way. Okay, you can just read it. Okay, now see here, there are something that we have to discuss here, because this is a little tricky, and the student feels some difficulty to understand the different variables, because there are three types of variable. So I'm going to give examples. Okay, based on the example, okay, you'll come to know that what are these type of variables and how do we identify. Because if you are going to identify the variables, you can make the proper graph. You can make the proper graph, either the bar graph or the line graph. Okay, so now come to the identifying the variables. So here, there are three types of variable. You can see here, like see, independent variables. See, independent, what is independent variable? First understand it. Independent variable is the variable that like you have to change it. Okay, you have to change it. And dependent, basically a dependent variable automatically changed by the independent variable. And something are kept constant. Okay, let me to tell you here about the activity of enzyme and the pH. If you are just finding the relationship between the pH and the active enzyme, or suppose if you are finding the effect of temperature on the active enzyme. Okay, so what the temperature is, temperature actually is in your hand. You are changing the temperature, and based on the temperature, okay, the active enzyme is being changed. So what is independent variable here? Temperature is the independent variable, because you are changing that you like you observe the chemical reactions, okay, in different temperature. Okay, you fix the temperature. So this is independent variable. And based on the changing of the temperature, what change take place in the active enzyme, either the active enzyme increasing or decreasing. For example, like if you are increasing the temperature from 0 to 37, okay, you will see that like as you just move from 0 to 10, the active enzyme increases. 10 to 20, active enzyme increases. 20 to 30, active enzyme increases. And when you reach to the temperature 37, the enzyme activity is the highest. That is the optimum activity. And that's what in this temperature is called as the optimum temperature, means the temperature at which enzyme work best. Okay, so independent variable here, temperature, and active enzyme is dependent variable. And control, what are the control variable? Control variable means something during the experiment you keep constant. For example, if you are having the effect of temperature on the active enzyme, so you have to keep the pH constant. How do we keep the pH constant? We have to add some buffer solution in it, so that the pH is going to be kept constant in all the experiment, in all, like the samples. The pH is going to be constant. Also the quantity, yes, quantity of the reactants, quantity of the enzyme. Okay, they should be kept constant. Okay, so anything that we keep constant, okay, that we call as the control variable. One more like thing is there, that is control. We will discuss later on. Okay, now come to the next slides. Okay, where we have to discuss. Okay, yes, three kinds of variable you can see. Independent variable, something that is changed by the scientists. This is what I told you, that if you are changing something, okay, that is the independent variable. For example, if you are changing the temperature, that is the independent variable. If you are changing the pH, okay, of a substance, so it is the independent variable. And what is tested, what is manipulated, means changed, okay. What the change that is in your hand, okay. Now another thing, here is like, dependent variable. See here, something that might be affected by the change in the independent variable. Yes, when you change the temperature, the active enzyme also change. So active enzyme, dependent variable. Okay, what is observed, what is measured, means what you are observing, what changes you are observing, and what you are measuring. So this is the dependent variable. The data collected during the investigation, the numbers, the example, how tall the plant grew, how far the paper airplane flew. Okay, these are the particular example of dependent variable. Now come to the next one. Next slides. Yes, see here, hours studies for exam and grades on 300 points exams. See here on y-axis, you can see total, like 300 points are there, and here hours studies, how many hours are studied. Okay, you can see here, if like the student is going to study 50 hours, okay, so what is like the grades point. Okay, so see here, it might be almost 50, okay. In the same way, see here, as the number of hours of studies increased during exam, then the grades on point are also increased. You can see here, this type of relationship, in which both the thing are increasing, we call as a directly proportional. Directly proportional mean, if you increase one thing, the other thing also increases. If you decrease one thing, the other thing also decreases. So this is we call as a directly proportional. And in inversely proportional and directly proportional mean, if you increase one thing, the other thing is going to decrease. So this is called as a indirectly proportional or inversely proportional. Now come to the next one. See, control variable. As I've told you, the control variable are those variable which you have to keep constant. Okay, if you want to, like the proper result and the correct data, so you need to control something. Okay, for example, if you are studying the effect of temperature on the active enzyme, so pH should be kept constant. If you are going to change the pH, you are not going to get the correct data from your experiment. Okay, so it is important that in all the samples of the experiment, you need to keep something constant. If the constant are not to be maintained, then you cannot get the reliable data. Okay, so for reliability, you need to keep something constant. Okay, so see, everything in the experiment except for the independent variable should be kept constant. Okay, everything you have to keep constant, but independent variable, it has to be changed. As I've told you, the scientists change the independent variable. Okay, but the others, like control variable, okay, has to be kept constant, and the dependent variable is being changed, okay, automatically by due to the independent variable. Now what is the procedure? Yeah, procedure mean method, what method we are going to apply here. Okay, so this is also very important, because if your method, if your procedure is good and there is no chances of the error, okay, you are going to get the perfect data. So just give a detailed explanation of how you will conduct the experiment, test your hypothesis. Because you see, whatever, like the gas you have just imagine, okay, whatever the gas you find out to solve the problem. Okay, so you make a hypothesis. This hypothesis, actually, the reliability of the hypothesis depends on the procedure. If your procedure is good, if your procedure has no, like flaws, no errors, okay, then you are going to get the correct data, or based on the data, based on the data you can rectify the hypothesis. So but be clear about the variables, okay, elements you change versus your constant element that you do not change. Mean which one you are changing and which one you're not changing, which one is the independent variable, which one is dependent variable, and which one is the control variable. This is very important. If you are going to just uh, care about the variables, okay, your experiment would be good, okay, your result would be reliable, and your hypothesis is going to be rectified. Okay, so a control is the group that you use as a comparison to see if the change has occurred. Yes, we also have to keep one type of samples, okay, in the experiment that we do not see any change in it. Okay, that is the control. For example, see, in a medicine study, the group of people who do not get the medicine are the control group. Okay, see, for example, if we make the corona vaccine, okay, and we are giving this corona vaccine to some patient, okay, and we see the effect of that vaccine, okay, and one group of peoples, okay, we are not giving any vaccine. Okay, so the group of peoples to whom we are not giving any vaccine, okay, this is considered the control group. Okay, because that is out of our research. Okay, we just want to, with the help of control group, we just want to compare the things. Okay, because you see here, we are giving the vaccine to one person, okay, what's effects is going on in that person, okay, and the other person to whom we are not giving vaccine, okay, what's going on with him. Of course, we can easily differentiate, we can easily differentiate that what is the effect of vaccine. Now, be very specific about how you will measure results to prove or disprove your hypothesis. You should include a regular timetable for measuring the results or observing the projects, such as every hours, every day, every weeks. Yes, so we have to be very specific. We have to be very specific, okay, in our experiment, because the experiment is going to prove or disprove your hypothesis. Now, conclusions, as I've told you, the last step, is the conclusions. Okay, that you see what is the output, okay, of your whole research. What is the result? Okay, so conclusion, your results are finding based on data collected during the experiment. Yes, if your experiment have no error, okay, you are going to find the correct data, then your hypothesis is going to be rectified. Answer your problems purpose statement means conclusion, always answer all your problem and what do you want to achieve, you can get it. Okay, what does it all add up to? What is the value of your project? Okay, what is the value of your research? Okay, conclusions, in conclusion, you have to write down all these things. What further study do you recommend given the result of your experiment? If something, if something you couldn't find out, okay, or for example, when you perform one experiment, okay, so there are two or three divergence, okay, one thing is proved but other thing also need to be researched, okay, after that it can be proved. So by the conclusions, okay, you can also mention the further study do you recommend given the result of experiment. What would be the next questions to ask? If you repeat this project, what would you change? What would you change? If suppose that you are repeating the experiment, okay, what do you change? Okay, what the new things appears? Okay, so this is also very important. So in conclusions, we have to write down all the aspects of your experiment and your hypothesis. Now, another example, see here, an investigation was done with an electromagnetic system made from the battery and wire wrapped around a nail. Different size of nails were used. The number of paper clips the electromagnetic could pick up was measured. So see here, we are just we take the nails, okay, and on the nails we just wrap the paper clip, okay, on the magnet, and then we measured the current. Okay, now see here, what are the variables here? See, the variable, as I've told you, variable are very important, and you have to be very much like, careful, okay, when you are just identify the variables. So see here, independent variable, is size of the nails. Okay, size of the nail is an independent variable, and dependent variable, number of paper clips picked up. Okay, so this is, and what about the constant? Yes, which thing you have to keep constant in all experiment, in all sample? The battery, white type of nails, none of these items were changed. Means you cannot change, like, the different voltage of battery in different sample of experiment. Even you cannot change the wire. Okay, for example, if for one experiment you are taking, like, you are using the copper wire, the other experiment you are using the silver wire, no, you cannot get the proper, like, data. You cannot get the reliable data. Now another example, let's practice. If you use a if I use a heavier bowling ball, then the ball will travel faster down the lane. Okay, means if you are going to just, like, use more power, okay, and you are having, like, the heavier bowling ball, then the ball will travel faster down the lane. Okay, but if you are having a light ball, okay, so you can see that what would be the result. Okay, now see, independent variable, weight of the bowling ball. Because independent variable, is why we say independent variable, because we are changing. Okay, either we are using the heavier bowling ball or the lighter bowling ball. And then dependent variable is speed at it travel downland. Okay, what is the speed that is required here? Okay, now if I use a different brands of paper towels, then Bounty will absorb more water per minute than the sparkles or quilted. Okay, this is quite logical. Okay, now come in independent variable, brand of paper towel, and dependent variable, amount of water absorbed per minute. Now I think this is the last example. Yeah, if I put three spider plants in different location, then the plants in the sunlight will grow taller in 1 week period than the plants in the closest and basement. See, if I'm going to the plant, okay, and if I just change the location of the plant, okay, because change the location of the plant, of course, like, the plant is going to get different amount of light. Okay, so you know very well that if the plant getting more light, okay, so more photosynthesis would be there. More photosynthesis means the plant is grow faster. Okay, so independent variable is the location of the plant. So this variable in my hand. Okay, either I'm going to just keep this plant in my in my room, or at the window, okay, or outside the house. Okay, so depends, like, what is the location. Okay, what is the location? So location change, okay, of course, like, the dependent variable will also change. Okay, for example, if I keep the plant in the lighted area, I will see the plant grow faster. Means dependent variable, height of the plant is going to increase faster. Okay, so dependent variable basically it depends on the independent variable. Okay, so now this is, you see here, this is all about, like, the scientific method. Okay, thank you very much for listening and your patience. Okay, if you have any questions, you may ask me. Okay, you can see here my email address, na.ali.siddiqui@gmail.com. And if you have further inquiry, you can also WhatsApp me if you have any questions. Okay, so thank you very much. Okay, I hope you enjoyed this lecture. Okay, and I hope that you understood each and everything. Still, if you have problem, okay, you can discuss me. Okay, you can email me. Okay, so thank you very much.